Okay, so as Tanya said, the schedule changed just a little bit. I'm clearly not Henning Tiemeier. If you're looking at the printed version, he unfortunately couldn't be here today. What I'm going to go over is very basic epidemiology, just to give everybody a bit of an overview as to what are we talking about when we're talking about applying epidemiology to human brain mapping. We, of course, can't cover everything under the sun in terms of epidemiology, so it will be a rough overview. So by the end of this talk, I hope that we have a better understanding of some basic epi principles, a better or more clear grasp on terminology and why terminology I think is important, and also a bit of an awareness of some really common misconceptions about epidemiology. So Tanya already went over, I think, roughly the same definition, I hope the same definition. So I won't spend too much time on this. Really simply, the study of the frequency and pattern of health events, or a professor that I worked with in Rotterdam who taught basic epidemiology called it occurology, or the study of occurrences. So we're all very familiar with how epidemiology is applied, for example, as Tanya said, in tracking disease states such as Ebola virus or something like that. But the reason we're here today to talk about epi is that we think there's some very interesting applications in human brain mapping. Now, epidemiology has, let's say, three main categories of how research is done or how the studies are done. And the first very obvious one is what we would call a descriptive study. So what does that mean? Well, a clear example might be, say, comparing men versus women in terms of their different brain features they have, or maybe just very descriptively describing a particular phenotype, a particular disease that you're interested in studying. Another type of work that we do in epidemiology is prediction work. Now, that's different than having a simple predictor in, say, a regression model, but rather trying to predict the risk that somebody is going to develop a particular disease or the risk somebody will develop a particular event. So a heart attack, Alzheimer's disease, and so on. Now, you're probably most familiar with prediction work if you imagine yourself going to the physician office, being evaluated for, let's say, a heart attack, myocardial infarction. You'll be asked a series of questions. What's your age, gender, do you smoke, do you have diabetes, and so on and so forth. And the physician has a lookup table, more or less, that can evaluate these different risk factors that you've sort of answered questions on and gives you a 1, 2, 3, 5, or 10-year risk estimate as to how likely it is that you'll develop whatever disease that they're looking at. In the third category is causal inference, or etiological research, we call it. Now, it vastly depends on the kind of questions and so on that you're asking in your work, but I would guess that a substantial portion of everybody here today is doing causal inference or etiological research based on the types of models and the types of analyses they're running. You may or may not be aware of this, but hopefully by the end of the session I'll convince you that that is the case. In particular, epidemiology has a very focused set of methodologies that they apply. And in the next talk by Anna-Marie, you'll see some clear examples as to why this is important and how it can help human brain mapping, and particularly in the context of confounded models and adjusting for different kinds of confounders. So this is another particular subfield of epidemiology. This is focused both in causal inference and in prediction. And then validity, everybody who thinks about a huge population-based study maybe is thinking, I have high validity, I have generalizability, I have huge power, and we're going to talk a little bit more today about whether or not that's always the case. Probably some of you are asking yourselves, how is this different from what I'm doing already? And that's a good question because actually there's a lot of overlap and there's a lot of similarities. What I hope you'll see again in Anna-Marie's talk next is that there are some subtle but very important distinctions between how a lot of us in brain mapping are running our models or thinking about the models that we're running. And Anna-Marie is going to go over that more. One really clear example is in study design. So a large proportion of work here at OHBM is in relatively modest sample sizes, very often, let's say, cases and controls as an example. and matched cases and controls and fairly minimally adjusted statistical models. And in large cohorts, like as Tanya was saying, UK-Biobank, we don't do a lot of case control matching, but more often we do things like adjusting for every possible confounder that we've measured. And again, there's some very obvious differences in terminology, which I think are really important for a number of reasons, just getting things right, helping reviewers review your papers, and also helping your audience understand what it is exactly that you're doing. So a couple of easy examples here. In most of the work that I did, I studied psychology. Most of the work that I did prior to getting involved in epidemiology, we thought of things as independent variables and dependent variables. In epidemiology, there's slightly different terminology, exposure and outcome in particular. They're relatively broad, relatively similar. The main thing that I think is important about terminology that I want to highlight here is that whatever terminology you use has to be defined properly. And if it's not defined properly, there's going to cause a lot of confusion in whoever's sort of reading your papers or looking at your work. And as I said before, there's one field of epidemiology called prediction work, which can get very confusing if you're using terminology like a predictor in your model, which isn't properly defined. Other terminology, covariate, is something very often used in our work. In epidemiology, we call this very often a confounder or a confounding factor or confounding bias. And then mediator and effect modifier are other terms that are going to be talked about by Anna-Marie in the next session. So in terms of validity, which I mentioned on one of the previous slides, there's, of course, two types of validity. This is very basic. Internal validity, dealing with measurement error, for example, and whether or not the study is internally valid, that there's free of as much bias as possible. And external validity, meaning whether or not the results of your study can apply to other populations, if they're generalizable to another setting. So one, I think, common misconception about big data is that if you have enough data, your validity is increasing with that. And in the next couple of slides, I want to go through, is that actually the case? Is it possible that having a big enough study solves all of our problems? So everybody's seen the bullseye, the dartboard, where you throw darts at it, and if you have a nice cluster of darts on the bullseye, that means that you have high accuracy and high precision. And so in the figure in the middle, you can see the red diamond, which is what our true or sort of underlying effect is if we happen to know what it was. And if we have a study that has high accuracy and high precision, there's a black diamond just below that showing that our high accuracy, with high accuracy, we get an estimate or a parameter estimate from our statistical models that's very close to what the underlying true estimate is actually is. With the black line, that's the estimate of the confidence we have around that estimate. So in this case, it's a relatively narrow confidence around that estimate. That's a reflection of the precision, essentially the sample size that we have. Now, the next situation is if we lose that precision, if we have a smaller sample size, but we maintain the accuracy, we still have an effect estimate or a parameter estimate that's really close to what we think the ground truth is. But the confidence interval around that is a bit wider now. So we've lost the precision, we lost the sample size. The interesting thing here is we get the same answer with a big sample size or with a small sample size. We just have less confidence around what that parameter estimate is. Now, the next situation that we can have if we have a large sample size, so if we have lots of precision but low accuracy, we end up with a situation of a biased estimate that's not reflective of what the true underlying estimate is. So we have a situation of an overestimate on the right side and an underestimate of what the true effect is. It has a small confidence interval around it, but more or less all you're measuring is the amount of bias you have in the data set. You're not measuring the true effect that you sort of think you're measuring, let's say. And then the worst case scenario that we can have is low accuracy and low precision, a large confidence interval around an effect estimate that's highly skewed to one side. It could be an overestimation or an underestimation. So here I hope that I've shown you having a huge sample like UK-Biobank is only so helpful if we're looking at an unbiased effect estimate. Or there are situations where, for example, in prediction work, where that's not necessarily a requisite, but we can get accurate effect estimates or parameter estimates from smaller samples. That's not always the problem. It's more about ensuring that we have unbiased estimates. So just a quick quiz. If you were to have to choose between having high accuracy or having high precision, I'm wondering what you might choose. I think there are situations where you may want to have high precision, but for me personally, if I think about a real life scenario here, and we're running lots and lots of, let's say, smaller studies, and we have a situation like is on the screen with high accuracy but low precision, at least if we meta-analyze these data, we can increase our precision that way. But if we have a biased effect estimate, running meta-analyses doesn't get us all that far, depending on the circumstances, of course. So I think, indeed, it's better not to jump to needing a massive data set to answer a question, as long as you have an unbiased effect estimate. As Tanya also just showed on her slides, Steve Smith and Tom Nichols have published a really interesting paper, Statistical Challenges in Big Data, Human Neuroimaging. I can highly recommend reading that paper. It covers a lot of the stuff that you're going to see in the session today. A little bit more topical than what we have in this session, but still very interesting, very worth your time. It's only a couple of pages. And there's another study on the bottom, recently published in Neuroimaging. This takes another angle, more or less forgetting about how generalizable your data are, but looking into how precise you can measure your data. Not necessarily with a large sample size, but instead precision measuring your say imaging data highly with high precision. So two papers that I would highly recommend if this topic is interesting to you. They cover more than just the validity things I'm talking about now. They cover a wide array of topics. So as Tanya mentioned before, there's a number of different kinds of bias that you can encounter when looking at these, well, not just in large cohort studies, but actually in every study that we do. One particularly important piece or important type of bias is called selection bias. This is a situation where the individuals either being selected into the study or individuals that you're following over time are leaving the study in a way that's related to the variables that you're interested in. So, as I said in the terminology before, exposure and outcome, or independent variable, dependent variable, however you want to call it, your selection or recruitment of your subjects is not random with regard to your study population or the population of interest. And it's also associated with the variables that you're studying. So this can lead to issues, namely that you have biased effect estimates like the ones I showed in the previous slide, the validity slides before. So as an example, study entrance, there are a number of different examples of this, but you can imagine individuals who, let's say, have higher SES was an example that Tanya mentioned, I think, and that Steve Smith mentions in the UK-Biobank paper that was written recently. Within my work in the Generation R study, a pediatric cohort, we have what we would call a selective loss to follow-up. So when the children entered the study, it was very representative of the catchment area, the population that we were interested in looking at. However, over the years, the study has changed because we're not. There are individuals who are dropping out of the study, and it's a non-random dropout that we have. So, as an example, our IQ, which is a measured with a mean of 100 and a standard deviation of 15, was exactly that at the onset of the study. However, now it's a few points higher because we're selectively retaining individuals with higher SES and higher IQ in the sample. Another type of bias next to selection bias is called information bias. There's a lot of different forms of this and equally important to think about in your studies. Recall bias is one example. It has to do with whether or not the information you're collecting from your participants is skewed or not, or is biased. So recall bias, one example, if you're polling, let's say, pregnant women who had complications at birth, if you're asking them questions about that process or how their childbirth went, they may selectively remember things better than women who didn't have complications just because the event was, they experienced the event a bit differently. That's a form of recall bias. Reporter bias has to do with how you're, another aspect of how you're obtaining information. Again, back to my work in the Generation R study, the mothers are answering information about how the psychiatric symptoms or the behavioral problems that their children experience. So you can imagine that if a mother herself has a particular set of psychiatric symptoms, let's say depression or maybe an eating disorder issues, she may report differently on her child's problems. So this is another type of bias that's really important to think about. And rater bias is probably one that most people here are very familiar with. So if you have your imaging data and you're, let's say, identifying a region of interest, the rater, whoever is defining that region, may be doing it systematically in a different way than another rater, as an example. So what can you do about this? It's challenging. There are some methods for dealing with bias. However, the best thing to do is to avoid it from the onset of your study. That's the only way to guarantee that it's not present. So ensuring that the selection is happening in as random way as possible with regard to your study sample. And then also trying to make sure that the way you're collecting your information is as standardized as possible. Now, another thing that I think as an organization that we could benefit from is understanding what does the bias do to our data. So there are two potential options. The bias that you get from selection bias or information bias or any other type of bias that we talk about, it can send our effect estimates upwards, so we can have an overestimate of what the true association is. It can also send it back towards the null, so we can have an underestimate of what the true association is. Now, one area where we can benefit is just talking about it in our discussion sections as an example. So mention what types of bias might be present, think about what kinds of bias might be present in your work, and mention it in your studies. Mention whether or not you think you have an inflation of your effect estimates or whether or not you have an underestimation of your effect estimates. So a quick question. I think I already gave the answer away, but does selection bias or information bias always end up in being an overestimation of your true effect estimate? True or false? Well, it's false. There are situations where you can actually predict what's going to happen, depending on how random, say, the information bias is. That will, in general, lead to an underestimation of the effect. If it's a systematic or a non-differential type of bias, then in general it may bias your study in the other direction, or it's a bit more unpredictable what might happen. So, not to get into Anna-Marie's talk, because you're going to hear a lot about confounding in the next 45 minutes. But confounding, or as I said before, we often call this a covariate in our analyses. This is a variable that's related to both your exposure, or your independent variable, and your outcome, your dependent variable. And it's if you think about it in terms of a causal diagram, it's not something that's an intermediate factor in this association. Now, the easiest example of this in brain mapping is motion. Of course, motion is going to be related to our structural imaging, to our functional imaging, diffusion imaging, and so on. And it's also going to be related to some of our phenotypes that we're interested in. So if you think about ADHD, individuals with ADHD are going to move more, and they're going to have biased structural or diffusion or functional features coming out of the imaging data. And again, more from Anna-Marie. At the beginning of the talk, I mentioned that there are three types of studies that we do in epidemiology. A descriptive study, again, comparing, let's say, just describing different features of a phenotype. So describing the different imaging features of depression, describing the different imaging features of Alzheimer's, describing differences between men and women. Prediction work, so building risk estimates of how likely it is that someone is going to develop a particular disease or an event. And causal or etiological work. Now, one important part of these different themes of research that I think is worth mentioning is that we're a bit, as a field in general, a bit messy with how we describe these things in our studies. So, if you were to pull a given paper from the literature, you may not know exactly what the goal of the study was or what kind of work they're trying to do. It may be difficult to assign one of these three categories. Because there's a bit of a mix of methodologies applied and a mix of terminologies that are being used. So I think it's important to be very explicit about what kind of work you're doing, to name it in your studies. In particular, if you're talking about area under the curve, you're talking about statistics and so on, you're probably doing prediction work. If you're talking about effect estimates or parameter estimates, and you're adjusting for covariates and you're looking at confidence intervals and so on, you're probably doing causal inference or etiological research. Now, another paper that I would highly recommend that folks pull and read is one by Miguel Hernan. It's called The C Word, Causal. This paper goes into great lengths describing why we're too afraid to talk about causal inferences. We very often call our work association studies because, of course, correlation does not imply causation. But the arguments made in this paper, I think, are very interesting, and there are two main points or main summary points that I'd like to say about it. One is Miguel very nicely notes that we shouldn't be afraid to use causal language, mostly because in many of the studies we're doing, the moment that we enter in a covariate and we're with the sole purpose of adjusting the model that we're running for that covariate and looking at the effect estimate, is it a strong effect, is it a weak effect? The moment that we do that, we're already doing causal inference. That changes, the story changes, landscape changes a bit if we're doing prediction work where we don't really care how the different covariates or confounders we add into the model, we don't care how that affects the parameter estimates. But if we're really interested in the association between A and B, we're doing causal inference as soon as we start adjusting for covariates. So that's the first point that I think is really interesting from this work. The other point from this work is that we should be very explicit, again, as I've said in other areas of this talk, about what kind of work we're doing. Define the different variables that we're using very well, define what's an exposure, what's an outcome, and define what kind of work we're doing, whether it's prediction, descriptive, or etiological work. Last slide here, before I pass on to Anna-Marie. We, just like in genomics, are very reliant on p-values, and this is going to be a recurring theme today, talking about p-values a bit more. In epidemiology, if you talk to a serious methodologist in epidemiology, and you want to talk to them about how should I adjust for multiple testing, what should I do with my p-values, and so on, the clear answer you're going to get from them almost every time is don't test, don't do inference testing. And that's a bit laughable here because, of course, we have to do some adjustments for multiple testing. There's not really any way around it. But in epidemiology, they have a slightly more simple mindset about things in the sense of we want to look at what's the effect estimate, what's the parameter estimate, how strong is the association, and what's the confidence that we have in that. So they disregard p-values almost entirely, as much as they can. So I think one interesting challenge for brain mapping as a whole is to start trying to adopt some of these techniques where instead of just focusing on which p-values are significant or not, that we start looking at parameter estimates and confidence intervals around those p-values, come up with, for example, neat ways of displaying these things. And again, as Tanya mentioned already, when we have sample sizes like UK-Biobank, this becomes even more crucial to incorporate into our work. So just a general recap, adding precision or adding sample size, it's not always going to be something that's helpful. If we have a biased estimate, we're just simply adding precision to what our bias, or measuring what our bias is. It's important to use consistent and well-defined terminology. This is for your audience. This is for reviewers. It will make everyone's lives easier to understand exactly what's being done in the work. With smaller studies, often discount potential biases. That can't be done with small studies, and it certainly can't be done with larger studies. So think about the types of selection bias that you have in your work. Think about different types of information bias. Those are the two crucial ones that you should be aware of. And just wrapping up, back to the Miguel Hernan paper about the C word, try to be as explicit as you can about the goals of your work. Are you doing a descriptive study? Are you doing a prediction study? Are you looking at causal inference? Again, it makes everyone's lives easier if you're very explicit about what kind of study you're doing. And it helps, for example, a reviewer evaluate whether or not you're applying your methodology, applying your models properly. Really quick, for those of you who are interested, these are sort of a brief citation of the 3 papers that I showed. Tom Nichols' best practices. Oh, sorry, I didn't mention the Tom Nichols' best practices paper. This is the OHBM COBITUS report from a couple years back. Definitely read it if you haven't read it already. There's a ton of good information in there, in particular from the EPI standpoint on how to report your findings. Steve Smith's big data paper, and then Miguel Hernan's the C-word paper. As I mentioned before, I'm not Henning Tiemeier, but he was originally planned to be here, and we also had a tremendous amount of help from these other individuals, Micah Vernoy, Arfan Ikram, and Henning Tiemeier. And that's the end of my talk. Thank you.